Good afternoon, everyone, and welcome. We'll get started in just a moment, but a few details before we get underway. We are recording the event, and the video will be available for you to watch on the Basic Science website. We'll post the URL in the chat during the event. You'll be able to see this video and other videos from past events this season and prior seasons. Due to the large number of people we're expecting to attend today, we are going to keep the audience muted throughout the event, but we do want to hear from you. So if you have questions for our panelists or comments that you'd like to share, we ask you to use the chat to post those. We'll be monitoring that throughout the event, and we'll try to get to as many of your questions as we can during the hour. And without further ado, it's my pleasure to introduce the Dean of Biological Sciences, Mike Bachman.
Good evening, and welcome to another installment of Basic Science Lights the Way. I'm Mike Bachman, and I'm so excited to be here this evening to see you all and share more fascinating science stories with you. As many of you know, we've launched this series about curiosity-driven science at Berkeley in the height of the pandemic. Though we are likely emerging from that, this series will continue. It's been growing in interest, and as a dean, I'm so happy to continue showcasing the important work of our many amazing faculty and students. Though this is our last event for this academic year, we will see you again, I hope, in the fall. The theme of this series, as our regulars know, is basic science research, also called pure or fundamental research. We attempt to show in this series why it is so important and why it is so central to Berkeley. It's through this type of open-ended research that we can refine scientific theories and better understand natural phenomena. Sometimes a scientist doesn't know where her curiosity is taking her. And those are often the most interesting and most fruitful directions. So yes, discovery and subsequent translations are the result of basic research. And that's why we often point to things such as CRISPR-Cas9 or the acceleration of the universe's expansion as outcomes for such commitments, and emphasize that such amazing work results from a long series of questions and experimentation that got the scientists to that point. And from there, you go on to learn more, a never-ending process. Once we get underway, we encourage you to post your questions for any of our speakers in the chat box. We'll answer as many of them as we can. Tonight's topic is science at the extremes. This is an especially fun topic, I hope, because you're going to hear about how some of our scientists deal with literal extremes in the lab and outside, thinking about handling dangerous viruses, doing science at high altitudes, or at the edge of a volcano. Our moderator this evening is Dr. Julia Schaletzky, the Executive Director of the Henry Wheeler Center for Emerging and Neglected Diseases. She's also the head of our Drug Discovery Center and the Immunotherapy and Vaccine Research Institute. Julia's interest and career spans pharmaceutical development, bioentrepreneurship, biotechnology, health policy, public health, and innovation. Over to you, Julia.
Thank you, Mike. I'm very excited to be here and to participate in tonight's event, and I'm thrilled to introduce to this audience three stellar researchers and their work. Haven't we all witnessed science at the extremes during the last year of the pandemic? This drove home the point like no other, that we can build on the foundation of high-quality basic research in order to meet the challenges we are facing. For example, mRNA vaccines coming to the rescue after 20 years of basic research that enabled their use. As our world is changing, scientific understanding can inform good decision-making and benefit all. I have long been impressed by Berkeley researchers who work with dedication, creativity, and rigor on some of the most exciting questions about the natural world. Today, we're going to hear from three inspiring scientists, Britt Glaunsinger, Stephen Self, and Adrian Lee. Britt works on viruses, Stephen on volcanoes, and Adrian on the cosmos. Talk about a wild ride. I hope you will enjoy these fascinating presentations. At the end of each talk, we will try to address all of the questions from the audience, so please add questions in the chat. Our first speaker is Britt Glaunsinger, a professor and associate chair of plant and microbial biology and molecular and cell biology. As a biochemist, her research interests encompass virology, gene expression, and the herpes virus. Specifically, her lab studies the creative strategies viruses use to manipulate gene expression in host cells and the viral factors that directly target RNA, and revealing how virus host interactions can have unexpected consequences for gene expressions that provide insight into both virus and host biology. Britt, what can you share with us about your research?
Thank you, Julia, for that wonderful introduction, and thanks to everyone for being here tonight. So, as Julia said, I'm a virologist, and while my lab mostly studies herpes viruses, over the past couple of years, it's probably not surprising that we have also transitioned to studying coronavirus biology. So tonight, of course, we're going to be talking about the science of extremes, as was described both in terms of size and destructive power, including topics like volcanoes in outer space, which it turns out this picture is not actually of. It's a picture of a sneeze. And nowadays, anytime probably any of us see a sneeze or hear a sneeze, it's going to conjure up some images of viruses. Now, in terms of numerical size, viruses are certainly also extreme. For example, there are about 100 million times more viruses on Earth than stars in the universe. And even in just this one droplet of a sneeze, in theory, it could fit about 2.5 million coronaviruses in that droplet. While that number is just a theoretical one based on volume, viruses like coronaviruses can actually amplify to staggering numbers within us. To do so, though, they need to hijack our cells' machinery and subvert our immune system. How they accomplish this at the molecular level is what my lab studies, because if we understand the details of the takeover, we think we have a good chance of being able to design therapeutics to stop it. Now, to study viruses like coronaviruses in action, you actually usually need a special type of biosafety lab that allows for work with airborne transmissible pathogens. And this lab is called a biosafety level 3 laboratory. Here I'm showing a picture of some of the researchers who work in the biosafety level 3 lab at UC Berkeley on things like coronavirus. As you can tell from this picture, the individual researchers basically have to have these specialized suits that enable them to have their own filtered air supply. And some of these sort of extreme safety measures can make the research there a little bit challenging. So the good news is that actually we can take individual pieces of the virus out of the context of the virus as a whole and study them much more easily and safely in what's called a biosafety level 2 lab, which is the type of lab that mine is. And there are a couple of pictures of it here. You can see 2 of my students working either with isolated proteins or RNA that have been taken out of the virus. That's Leah Goulias here at her lab bench. Or with human cells that have components of the virus in them in isolation that are not infectious. This is Michael Lee, another PhD student, and he's working in what's called a biosafety cabinet on those cell lines. So we do our research in this type of lab, which enables us to answer really detailed questions about how individual viral genes are expressed and operate, as I said, with the goal of figuring out how we might design ways to stop them. So to do that, I want to step back and give you a brief primer on gene expression in our cells, which may be a reminder for some of you. As you probably remember, our own genes are encoded within our genome that is made up of double-stranded DNA in cells. We've got about 20,000 or so genes in our own human genome. And for those genes to be expressed, they first have to be converted from this double-stranded DNA form into a form of a molecule called a messenger RNA. And this is a molecule that has basically the set of instructions in a form that can be read by the protein making machine of the cell, which is called the ribosome. And it converts those messages into proteins, which do the work of basically everything that our cells need to have done in them. Viruses like us also have sets of genetic information. It turns out that coronaviruses, they're a step closer to making proteins than we are because their genome is this messenger RNA itself, but it's a lot smaller than ours. They only have 13 genes compared to our 20,000. And so that means that while they have some of the things that they need in order to amplify, they actually can't encode everything. They're just too small. And one of the critical things they can't encode is this protein making machine. They just don't have enough space in their genome. So they need this protein making machine, the ribosome. But another challenge for viruses is that during infection, one of the key activities of the ribosome is to make antiviral proteins that are going to activate the immune system and shut down that infection. So on the one hand, the ribosome is a piece of machinery that the virus absolutely has to have. But on the other hand, it's a source of proteins that are toxic for the virus. So my lab has been working towards understanding how coronaviruses can overcome these problems and how they do it using a viral factor that's thought to play a really important role in the ability of these viruses to cause disease or pathogenesis. So a central player in the story that I'm going to tell you today is a portion of that ribosome protein making machine. It's called the 40S subunit or the 40S ribosome. In this ribosomal subunit, there's a channel or a tunnel that basically fits that set of instructions inside of it in order to read them and create protein. Well, coronaviruses have a protein that's called NSP1. It's like a molecular cork that inserts right into that channel, stoppering it up. And so that when cellular messages, very importantly, like those messages with instructions for making antiviral proteins that boost the immune system, when they come to be made, they can't be translated. So this solves one of the very important problems for the virus. In shutting down the ribosome in this way, it can block the ability of the cell to mount an effective immune response. And in fact, it's been shown that if a coronavirus doesn't have this protein, the virus has a very hard time amplifying well in cells because the cells basically stop it using the innate immune system. All right, so that's one solution. It's almost like thinking about the virus robbing Peter to pay Paul, so to speak, though, because if it blocks the ribosome, coronavirus can't get its own proteins made either, because remember, it needs this machine. However, it turns out that the viral messages have a way around this. All of the coronavirus messages, messenger RNAs, have a shared sequence in front of each gene. It's called a leader sequence. And this sequence gives them special access to the ribosome. So we think what happens is that when the coronavirus genes come upon a ribosome that has that sort of molecular stopper in it, they cause this NSP1 protein, the viral protein that's blocking access, they cause it to shapeshift in a way that removes that cork-like activity out of the ribosome and allows the viral messages in. So what can we do with that information? Well, one of the directions that we've taken is to try and identify what specific regions of NSP1 are critical for repressing cellular translation and allowing viral escape, because that might provide us with clues for how to target this protein therapeutically. So to do this, we've made many different mutations in NSP1 that's indicated here in these red dots. And then we test each of these mutations to see which ones might, say, functionally break the protein. Surprisingly, we found mutations that didn't just break NSP1, but in fact, instead reversed its targeting. So you can see that in this graph here, where what we're doing is we're measuring the ability of a cell to produce a particular protein. And so at 1 means 100% protein is produced. And if you can see here that if we're looking at the ability of a cell to produce a transcript that would be a cellular transcript, in the presence of that stopper, that translation is significantly reduced. But if you have that special coronavirus protective sequence on it shown here, well, then in the presence of NSP1, this message escapes repression. But now if we look at the mutants we've made, what we can see is that we can generate mutants that are now no longer very effectively repressed by NSP1 if they're cellular genes, but if they have the viral special sequence on them, NSP1 actually gains the ability to repress these. And so we're thinking about this as a way of converting NSP1 from a protein that helps the virus to a protein that is now antiviral, sort of turning it against itself. All right, so what we can do in the future is take this set of information and use it to screen, for example, for drugs that can bind these very critical regions that we've identified, perhaps in collaboration with Julia, for example, and be able to identify ways of stopping its function to fight the virus at large. I will end there and just acknowledge the set of researchers in my lab and others on campus that have participated in this, UC Berkeley, and other funders of our research. And I'm happy to take any questions. Thank you for listening.
Thank you so much, Britt, for a wonderful talk. And I just want to acknowledge also to you that, you know, you're one of the heroes of science. I think early in the pandemic, working in the biosafety level 3 lab with students on a virus that was so dangerous before the vaccine existed. I just want to point that out. You know, you really contributed to the campus response on this, and I'm very grateful for it. And I think scientists as you inspire us all. So we have some questions from the audience. One of them is, where does NSP1 come from, the virus or the cell?
Great question. NSP1 comes from the virus. The virus makes this protein. It is actually the very first protein that the virus makes as soon as it gets into the cell, which gives you a bit of a clue that it's playing an important role for the virus in stopping the cell from being able to mount an antiviral immune response, something the virus wants to set up really early in the cell as a way to escape that shutdown.
Any other questions from the audience? Don't be shy. This is a complicated field, so it's fine to ask any question. As people are thinking, yeah, do viruses compete with each other to prevent co-infection?
Excellent question. And indeed, they do compete with each other. And in fact, some viruses have ways of blocking other viruses from getting into the cell that they're in. So they can basically mark that cell as, nope, this is my territory. If a new virus were to come in and try and infect, they have a way of actually blockading those viruses from getting in. Not all viruses do this, but we know a lot of examples of ones where they're sort of actually competing with each other.
Okay, great answer. How are disease-causing viruses different from non-disease causing viruses?
Yeah, this is a really active area of research. What is it that will induce a certain virus to cause disease in humans? And there's a few different reasons why we think that the difference arises. One is that viruses that are not very well adapted to replicating in us tend to be really highly pathogenic. So when you think about viruses that are spilling over from an animal into a human, as we thought, as is likely how SARS coronavirus came to be, or other things like Ebola virus or Nipah virus or things like that, these because they're not yet well adapted to the human population, they tend to cause very severe disease in people, whereas viruses that have existed with us for long periods of time, herpes viruses, for example, can, for the most part, cause very mild disease. So this is not a hard and fast rule. It's not always the case, but that is one example of why a virus might cause more severe disease. Another reason is that sometimes causing disease benefits the virus. So for example, if it's a virus that is transmitted through airborne transmission, through you breathing and coughing, then a virus that induces you to cough and sneeze and produce a lot of sort of excrement is going to be, that is going to benefit the virus. So they will have evolved probably to maintain that type of disease.
And in that vein, how do you see this relationship between more aggressive virus infection and more infectious, transmissibility question and virulence?
Yeah, it's not always a perfect trade-off. And so often, if you are too virulent as a virus, you're not going to be transmitted very effectively. And this is something that we think about in the context of Ebola virus outbreaks and epidemics. That virus is so virulent that often it would sort of burn itself out, so to speak, before it could spread incredibly widely. Whereas a virus like CoV-2, which has now evolved to be very efficiently transmitted in humans, can do that because in most of the people that it infects, they're not going to die within 7 to 10 days of getting the virus. And so having high pathogenicity often comes at a trade-off for being able to transmit really widely.
And then some questions are really coming in. Thank you, audience.
Wonderful.
Wonderful. Some questions around NSP1. Have other non-COVID viruses used NSP1 blocking type of mechanism? And also NSP1, does it somehow interfere with the mRNA vaccine?
Love those questions. The first question, NSP1 is unique as far as we know in how it is stopping cellular gene expression. Actually, my lab's specialty is studying how different viruses block the ability of the cell that they're in to express their own genes. And so they have a number of strategies that we work on to do it. And the NSP1 strategy is a unique one in science so far that's been discovered. It's one of the reasons that we find it so fascinating and are interested in working on it. What was the second part of that question?
Does it interfere with the mRNA vaccine?
Oh, yeah. Nope, it doesn't interfere with the mRNA vaccine. In large part because the vaccine is going to get into a cell that is probably not infected with the virus in the first place. So, yeah.
All right. And then another question, are there beneficial viruses? Is everything negative, our interaction with viruses?
Most of the viruses that we know about are negative, but that's not probably because most of them are bad. It's just that it's harder to discover the ones that have beneficial interactions, and those are now coming to light in a number of really interesting ways. One of the ways is viruses that infect bacteria, for example. So you may be familiar with the fact that our gut and our GI system is chock full of bacteria, our microbiome, which controls lots of aspects of digestion and immunity. And the viruses that infect those bacteria actually can have either positive or negative impacts on the gut microbiome, and so could be beneficial for us. And we can also use viruses, develop them in ways so that we can convert them from potentially being pathogenic to being therapeutic. If you think about using viruses for things like immunotherapy or developing them as vaccine vectors or things like that. So we can actually convert them into doing useful things for us.
Great. And then a question from Jeremy. Does this virus encode an RNase that cleaves off the 7-methyl G-cap from cellular transcripts?
Not this one, but another one that we study called vaccinia virus does encode what's called a decapping enzyme that basically decapitates that protective sequence right at the 5' end. Coronaviruses do not. They encode their own enzyme that puts the cap on, but they don't have an enzyme that snips it off.
And last question, just folding two really big questions into one that we could talk about an hour each. How does the virus figure out the trade-off between virulence and transmissibility? And the other one is, how does the viral capsid assemble?
The trade-off question is just through evolution. Viruses can evolve with remarkable speed, as we've all sort of witnessed in real time through this pandemic. Every time that genome is being copied, and it's being copied trillions of times in each infection, you're generating a spectrum of mutants which are being tried out and selected for, depending on whether they can escape the immune system or be more transmissible. And so it's basically like a jackpot lottery of trying all different combinations, which they have the freedom to do because they mutate so rapidly. And how does the capsid assemble? It assembles once the virus makes the spike protein and has copied its RNA and then uses that RNA to sort of bud into internal membranes in the cell to capture that outer coating that protects its genome that has that spike protein on the surface, and then basically is released from those infecting cells. You can get a single input viral particle, and then that one cell will release hundreds and hundreds of new particles to go and spread and transmit to other cells.
Thank you so much, Britt, for these great responses. It's clearly a super interesting field. I've always been fascinated by virology.
Yeah, thanks everyone for the wonderful questions.
Bravo. Our next speaker is Stephen Self. I'm really excited to introduce him. He's an adjunct professor of Earth and Planetary Science. And I think it's fair to say where everyone is running from, he's running towards, because his research interests center around physical volcanology. And that is as exciting as it sounds. So field studies of products of large eruptions and the environmental impact of volcanism. With 40 years of experience in the study of volcanoes and volcanism, he has published numerous papers and articles. He also previously worked for the US Nuclear Regulatory Commission and has held numerous faculty positions. Tonight, Stephen will be sharing his experience as a volcanologist with us. Stephen, what would you like to tell us about your research?
In volcanology, I'm best known for the Volcanic Explosivity Index. It's got me a couple of thousand citations over the years.
Most natural phenomena have some scale or index, like earthquakes, allowing one to be able to compare the different sizes of the events.
This is known as the VEI, and it's the globally used measure to rate volcanic eruptions, the seriousness and the size of volcanic eruptions. Shown graphically on the screen, goes from 0, non-explosive eruptions, up to 8, well actually 9. It's an open-ended scale, from tiny eruptions like little burps on Mount St. Helens to the major 1980 Mount St. Helens eruption, which is moderate size eruptions, up to the biggest eruptions that the planet has. It's still currently used, amazingly, despite being proposed 40 years ago, as seen for the January this year, 2022, Tonga explosive eruption, which really surprised the volcanological world. There's a picture of Tonga erupting prior to this year. Very spectacular picture of an exploding volcano. And then the header on this article is the VEI 5 millennium eruption of Tonga. They've got a series of satellite images of this spectacular eruption cloud growing. It did, I think, January 15th or 16th of this year. So the next picture shows Chris Newhall, who's my co-proposer of the Volcanic Explosivity Index. He's shown in this picture. And he's the guy here that I'm pointing to. This is Chris, and he was a grad student when we worked on this project together to invent the VEI. And what we're doing here, we're at the caldera, the new hole in the top of Mount Pinatubo, which erupted in June 1991. And we're leading a field trip based on a conference there. And this picture was taken in the year 2000. So we've got a lot of happy, muddy people here up in the caldera of Pinatubo. The problem with the VEI, and I'm amazed, as I said, that it's still used 40 years after its proposal, is that when we proposed this, 1982, volcanology was in its infancy, and there was very little quantitative data available on eruptions. And this index is semi-quantitative at best, and it's really only based on 2 to 3 related criteria. One is the eruption column height. And in fact, what Tonga has just done in January has made this category or relating different volcanic column heights more uncertain. We still don't have a clear picture of what went on yet at Tonga, but it defied sort of our knowledge on which we've been predicting the heights that volcanic eruption clouds can reach. The other thing it's based on is the volume of the deposits of an eruption, which are very difficult to ascertain. You can imagine, as you've seen from the picture here, of a weak VEI2 eruption from Krakatoa in 1979. The deposits often are laid down in the ocean, in the sea, and very difficult to ascertain true values, realistic values for the products of an eruption. The duration of the eruption is related to this. Obviously, if an eruption lasts longer, the more magma is erupted. But once again, it's a difficult parameter for us to get a good handle on. A more powerful VEI eruption might look like this. Spectacular volcanic eruption column rising up above the tops of the clouds in the troposphere, rising up probably to the level of the tropopause between the stratosphere and the troposphere, and spreading out in this spectacular umbrella cloud, from which the main dispersal of ash takes place as it's blown along by the stratospheric winds. Here's another view of the same eruption, which is in Puyehue in June 2011. It's a volcano on the Chile-Argentinian border. It was rated VEI4, a sort of average medium-size eruption. It had a measured eruption column height here of about 15 kilometers. It actually did, I think, reach just above the tropopause into the stratosphere. But we've never had a big enough eruption since we've had cameras and satellites and things like that to get a view of what an eruption like that of Tambora in 1815 would be, which had a VEI of about 7 and a much higher eruption column. Various people have added to our index over the years, and we find other things tagged onto it, like was the stratospheric injection, the percentage of eruptions known and of this size in the last 100,000 years. And perhaps the thing that we understand least well for the really big eruptions is how often they occur on Earth. We really don't understand this very well at the high end of the VEI scale.
These extreme volcanic eruptions have been highlighted by having the VEI scale and have shown us a lack of knowledge about this large scale of volcanism.
Anyway, this is getting towards the end of my little presentation, but I had a kind of surprise when we did this. I found out that our next speaker, who's cosmic ray physicist Professor Adrian Lee, is going to make his measurements at a cosmic ray observatory in Chile. And this observatory happens to be situated on the edge of one of the biggest volcanoes on Earth,
known as La Pacana Caldera,
and one that I've studied in the field in the high Atacama Desert. Got a satellite image of it here to show you. The blue dots are where there's a regular astronomical observatory there, and there's going to be this cosmic ray observatory. They're situated here, and outlined in these orange lines is the huge volcano that sits there, one of the biggest volcanoes on Earth. It had a major eruption 4 million years ago, and it's had lots more recent activity. And this is an assemble of satellite images of the area. So you can see that the observatory sits on this kind of lump at the end of the northwest end of the volcano, on the edge of the volcano. And this lot of volcanic deposits here were produced by the latest eruption, from the latest big eruption from this volcano, which was about 1 million years ago. So the whole volcano is about 80 kilometers long and 35 to 40 kilometers wide. It's really an enormous thing, and the eruption that started it all is one of the largest eruptions that we know about. So it's going to be VEI 8 or 9 eruption, but as I said, 4 million years ago. Don't worry, La Picana is highly unlikely to erupt again in our lifetimes or even in the lifetimes of these planned observatories. So that's it.
Thank you, Stephen, for a wonderful talk. Really exciting. So I have a couple of questions already in the chat. One of them is, I used to live in Hilo, Hawaii, and witnessed Kilauea erupting about 8 times. How does it rate on your VEI scale?
I'm sorry, could you just repeat the question?
Yes. One of the audience members used to live in Hawaii, in Hilo, and witnessed Kilauea erupting about 8 times. How does it rate on your scale, on the VEI scale?
Well, Kilauea can have pretty explosive eruptions. And the one that was probably going on when the questioner was living there is probably a VEI 2 or 3, very small scale. But geological studies there on Hawaii have proven that it can produce eruptions up to about 4 or 5 on the VEI scale from the same volcano.
Great, thank you. And then another question, really interesting, where does the heat really come from that drives volcanic activity? Does it derive from radioactive decay or from gravitational tidal forces?
It's primordial heat stored in the earth from the accretion of the earth as one of the planets as the solar system was assembled, plus radioactive minerals and radioactive energy in the mantle of the earth, helping melt parts of the mantle to produce magma.
All right. What measurements would you propose adding to improve the VEI, and also why is there no component for the composition of the lava in the VEI?
Well, it's been shown, and interestingly, that almost any composition magma can produce a whole range of volcanic activity, from lava to very explosive. And so having a compositional component to the VEI wouldn't serve that much of a purpose. I think what I would like to do to add as a new component is something based on satellite images of the plumes, because we're now getting enough images to be able to build up a sort of worthwhile number to put into the index. Something about the spreading of those dynamic umbrella clouds would be very useful.
Excellent. And then a very interesting question from Richard about the Tonga eruption. How could it generate a tsunami wave in the Atlantic generated by the eruption pressure wave? How often does this happen? Does this relate to the VEI?
Indirectly, yes. It was first found out after the Krakatoa eruption in 1883 and described by the Royal Society of London report. And then, but this is the first recent, well, there wasn't an event like this in the 20th century. So this event produced an enormous shock wave. If you saw on the satellite images I showed, there's this circular wave moving out ahead of the main cloud. And this shock wave was actually physically tied to the sea surface as it swept around the Earth. And it disturbed the sea surface, which generated small tsunamis at the coastlines in some of the oceans. So this air wave, sea wave coupling has been known about since the Krakatoa eruption, but this is a lovely example of it that Unga Tonga produced.
Thank you. Thank you, Stephen, for great answers and a very interesting talk. So with that, I would like to introduce our final speaker, Adrian Lee. So going from the microcosm to the crust of the Earth, now to the macrocosm, out into cosmology and deep space. Adrian Lee is a professor of physics, and his research focuses on studying the cosmic microwave background. And if you don't know what that is, he will explain it to you. Adrian received his bachelor in physics and then went on to graduate from Stanford University, where he worked with Blas Cabrera on the early development of an experiment to detect non-baryonic dark matter. As a postdoctoral fellow at Stanford as well, Adrian then switched to biology and worked on mapping functions in the human brain using magnetic resonance imaging. He came to Berkeley to work with Paul Richards, measuring spatial anisotropy in the cosmic microwave background. He's a principal investigator of the Simmons Observatory, and he will share with us what it's like doing physics research in Chile's Atacama Desert and Andes Mountains. Over to you, Adrian.
Thanks very much for the nice introduction, and thanks to everyone for coming. So as you heard, I'm going to be talking about probing the extremely early universe with extremely small detectors from an extremely high site. So first, extremely early universe. This cartoon has a modest goal of explaining the entire history of the universe. Right now, all evidence points to the idea that the universe started with a bang. In fact, the Big Bang. And here's a fanciful picture cartoon with rays coming out and rings around the Big Bang. During the entire history of the universe has been expanding. That happens even at the very beginning. The universe comes from microscopic size, the visible universe today, from microscopic size, and is growing. Now, this Big Bang model, even though evidence points toward it, the simplest Big Bang model fails to explain some very simple things we observe about the universe. For example, the fact that anywhere you look in the universe, it appears to be the same temperature, a very cold 2.7 degrees Kelvin, 2.7 degrees above absolute zero. So to improve the Big Bang model, people have come up with this idea called inflation, and that's this era here. So what's happening there? Well, as the universe expands, it is cooling and getting larger. It's still very hot at this point. The temperature or the energy scale is so high that really everything that's happening is the domain of high energy physics. And in fact, gravity itself is acting quantum mechanically, which is not observed in everyday measurements. So during this very high energy period of the universe, this idea of inflation may have happened. And here we have to bring in Albert Einstein. Einstein told us that space itself acts as a malleable fabric. And in fact, during inflation, the idea is that the entire fabric of space is expanding exponentially. In fact, space is expanding faster than the speed of light. And that's not a violation of special relativity as it might sound, but it's a very dramatic event, early universe. And if this happened, a very small part of the universe is inflated exponentially, as I said, and that part of the universe goes on and continues to cool until about 400,000 years after the Big Bang. And that time is important because it's a time where the universe has cooled enough that it becomes transparent. Before this time, just like light can't get out from the center of the sun, light can't come out from this earlier time. But at 400,000 years, it's cooled enough that it's transparent, and we can see photons from this period all the way in our telescopes today. And what happens with inflation is it encodes a pattern of polarization light, polarized light, in that early era of the universe. And that's what we're looking for with our telescopes. Okay, so I have to keep going with the history of the universe here. So as the universe continues to cool, we have stars form, galaxies, clusters of galaxies, the Earth, and then finally the Democrats, Republicans, and our telescopes. So here we are at the very end, where we're looking back in time to this very early era where the universe became transparent and looking for this signature that inflation occurred. And if we could see it, it would really revolutionize our understanding of the universe and what happened at the very beginning. So the next part of my talk is the extremely small detectors. And why do we need these extremely small detectors? Well, let's go up to the right. This is the old way. This was the Planck satellite launched about 15 years ago. And it has only had 52 detectors, which was state of the art at the time. And you can see why it was hard to make. All of these parts are machined by hand assembled. Every function in this focal plane to receive light, and these are horn antennas, and then detectors in the back behind this plate. Everything's built by hand and hand assembled. So this is the new way, which we're working on now, where we're building things the way you would build a computer chip, the way you'd build the processor in your computer. And with the new way, we can put 1,000 to 10,000 detectors in the same area behind the telescope. So this is a huge jump for us in technology. People have likened it to Moore's law. And I'll zoom in successively here. So here we go to one silicon wafer. This is again the way you'd make a computer chip. This is about the size of your hand. Then I zoom in again to a single pixel, and this is 1 pixel in our camera. And the size scale here, this is a cartoon of a tip of a pen, a ballpoint pen. So this is 1 millimeter across here. And there's an antenna here that receives the light from the antenna, from the telescope, and then sends it on a wire where there are filters that pick the color of the light, and finally sensors that detect it. And that leads me to my last zoom in, where we get to the sensor itself, which is at the center here. And this is at the scale of 50 microns. I show a human hair cartoon here for scale. And the smallest feature here is 1 micron, which is a millionth of the size, a millionth of a meter in scale. And we build all this with lithography, as I said, the same way you make computer chips. And we use a facility on the UC Berkeley campus. It's relatively new. It's called the Marvell Nanofabrication. And here, just like in Britt's talk, we get to wear special outfits. But here, we're not protecting ourselves. We're protecting our detectors from us and our dusty hair, for example. So here I am with 2 of my former graduate students and an undergraduate student. Here are the bays where we do the work. And it's much like making an old style photograph. You use light to expose the patterns. And from this nice laboratory, you can actually see the Golden Gate Bridge. So it's actually quite a nice lab to do your work in. Okay. So my last topic is the extremely high site. And we're working in Chile. And here's a drone shot. And you can see here's a truck where we would commute to work. It takes about an hour. And after an hour driving, we get to 17,000 feet. And you can see, and connecting to Stephen's talk, this is this area where all that volcanism happened. And I was very glad to hear that it was 1 million years ago or so. We don't have to worry about next week it going off. Although we do see some peaks with smoke coming out of them. I'll have to talk to Stephen about that. So you can see some of the telescopes up here. I work on this one over here, which is called Simon's Array, and we're building a new one here called Simon's Observatory. And why do we go to such a high site? It's because water in the atmosphere absorbs the light from the very early universe, so it makes it 15 billion years across the universe, only to be absorbed by water molecules right above us. And so to avoid that, we get very high at this site, and you can see how dry it is from the lack of vegetation. It's one of the driest deserts in the world. And here's another movie where this shows one of the existing telescopes. It's this is 2 meters across. It's a parabolic dish. Light comes in, hits here, hits another mirror, and then there's a camera in here that receives the light. It has those detectors that I showed you the pictures of. So you can get a feel for how high and dry this site is. And it's high enough that the air is half the pressure that it is here in Berkeley. And it's fairly challenging to work up there. It's also quite cold because of the high altitude. You can see the snow there, and the snow will persist for quite a while because of the low temperature. Here's a picture of some of my group up there working. We're underneath the telescope. We're working on installing that camera with the detectors here in the back. So you can see everyone's wearing an oxygen tank. There's a tank on their backs, and it comes to a tube, and there's two little things that go, prongs that go into your nose. And every time you breathe in, it shoots a little bit of oxygen into your nose. And it keeps your IQ somewhere close to sea level values, but perhaps not quite. So we have to be extra careful up there. So putting it all together, we're on the hunt to understand what happened in the extremely early universe at this 10 to the minus 43 seconds. So that's a billionth of a billionth of a billionth of a billionth of a second of the age of the universe. And to do that, we need to make some extremely small detectors that have features that are 100 times smaller than a human hair. And we need to take them to the top of extremely high sites in Chile at 17,000 feet, where the air is very thin. And last slide, I just wanted to thank all the people who support our research that I discussed today. Thank you very much.
Thank you very much, Adrian, for a wonderful talk and really fascinating topic, working in such extreme environments, asking truly the big questions. So some questions from the chat for you. One technical questions, are the antennas tunable and individually tunable?
Yes, they are, and it's done by the way we designed them. To give people some background to the question, every antenna has a length that's close to the wavelength of the light that you want to receive. And for example, in your cell phone, there's an antenna that has the right length to receive the signals from the towers. So basically, we adjust the length of the elements in those antennas to select the wavelength of light we want to detect. And that's something my graduate students work on quite a bit, the cycle of designing those antennas.
Excellent. And then another question, how does the Webb telescope factor into your work? So the new infrared telescope Hubble upgrade.
Yes, it's very exciting that Webb went up there. And in fact, it's in the world of astrophysics, it's a little bit distant from what I do, but nonetheless, I woke up during the holidays to watch every day to see how it progressed. What they're after with the Webb telescope is understanding how the very first stars formed. And so that's later than this very early era, so close to the Big Bang. And after the cosmic microwave background photons are released. And part of that story was that in that very early time in the universe, there are already density fluctuations. They're very small though, they're 1 parts per million. So like waves on the ocean in terms of the height to the depth. And but those density fluctuations grow due to gravity. And so the regions that are dense get denser and denser and denser. And what happens eventually is you have conditions to form the very first stars. And that's where the James Webb telescope picks up. It wants to detect light from those very first stars.
Awesome. Thank you. I was really excited to see that delivering the first pictures. Another question about the microwave background, cosmic microwave background, the temperature density fluctuations seen within the CMB are said to have occurred during inflation. Do mathematical models exist that precisely fit the fluctuation levels seen?
Yes, and that you can, now this is a very important topic in my research. To see the fluctuations we've seen, they're consistent with inflation, but they do not prove inflation. If they had proven inflation, my talk would have been very different. And but to talk about the fluctuations and computer models, yes, one thing about the early universe that's really amazing, it's a very simple, I'll call it an object, or a very simple time in the universe, where you could describe the entire universe by a very very small number of numbers, the density, the temperature. And so the fluctuations are part of that. They're very easily describable. And in fact, they're really amenable to making a computer simulation that actually gives a very accurate imitation of what we see. And so that's key to our research. The fact that you can, with very simple physics in a computer program, predict what we'll see, and then we really do see it. So that gives us hope that if there is a signal from inflation, we'll really be able to prove it.
All right. And are you close to detecting circular polarization in the cosmic background?
Well, so let me say two things to that. The exact thing we're looking for is actually a linear polarization. So light can have linear or it can have circular polarization. Linear polarization is what we're looking for, but we're looking for swirling patterns of the linear polarization. So you think of little sticks as a graphic. They're the thing we're on the hunt for from inflation are swirling patterns. Okay. Now, are we close? Well, one thing about we don't know, because we don't know exactly how strong the signal is. And people have been looking, and we keep digging deeper and deeper, and it's no violation against the theory. The theory can allow a very wide range, and we're going to keep digging. So it could be that you open your newspaper in 6 months and there'll be a discovery, or maybe it'll be 6 years. But I hope we do get to discover it. But if there is a question about circular, that would be very exotic physics. No one predicts any circular polarization. And actually, it's worth looking for, because everything that people have thought of is very exotic physics. It will be very interesting.
All right. Thank you so much, Adrian. And thanks to the audience for very, very informed questions. You know, we're coming to a close. I just want to close out with one question to the group, to all our speakers. What are you most excited about in science at this moment today? Like, what really piqued your interest? We start with Adrian.
Boy, that's a tough question. I think that you know, connecting to Britt's talk, I think obviously all scientists have been watching what's going on in the world, and we look at it with our science, you know, brains. And in fact, one of my colleagues actually wrote a paper about the mathematical statistics of how COVID is propagating. And you know, so, you know, we're all, you know, focusing our brains on thinking about it, even though, you know, my exact work doesn't touch on that, or I don't have special, you know, abilities. But so I was really fascinated to hear Britt's talk and how that connects.
Thank you. And Britt next. Just briefly, we have to.
Yep, I'm just going to say that the thing that I guess I'm excited about is how many new people got brought into the things that I thought about as a virologist because of the pandemic, and have dramatically reshaped the ways in which we think about pandemics and think about addressing them. And so the vast sets of expertise, even connecting to, yeah, like the speakers, Adrian and Stephen, that we had tonight, I think it's just going to shape science in new and exciting ways.
Thank you, Britt. And Stephen? You're muted.
I'm very interested and concerned about getting young children into science careers, and getting them to realize as early as possible where the horizons are. Because mostly with the education system we have, they're not interfaced with this until quite late in their lives, when most of their development has gone. So it's a very unscientific thing, perhaps, but I think it's very important for the future of all of our careers and all of our sciences.
And thank you for doing this work today, having science in an approachable way for a general audience. I think that helps with that very problem. So thank you so much. To close, I'm going to hand the floor over to Richard Allen, Interim Dean of the Division of Mathematical and Physical Sciences. Richard, the floor is yours.
Thank you, Julia, and thank you to our speakers. Thanks to Britt, Stephen, and Adrian for sharing a glimpse into how you all manage to do research environments that can be somewhat dangerous or unpredictable. This was a really exciting group of talks, and I really enjoyed the discussion and seeing how the work in the laboratory can be as stressful as high altitude and low oxygen kind of environments. A really great connection there. If nothing else, I think it shows how we all sort of work, or how everybody is determined in science and driven to get the data that they need to answer these very fundamental questions that we've heard a little bit about today. It's been a real pleasure for me to participate in this Basic Science Lights the Way series as Interim Dean of the Division of Mathematical and Physical Sciences, and also as a speaker earlier in the year about earthquakes, another very unpredictable hazard, could be a few months or a few years, as Adrian just commented for his science as well. I've completely enjoyed learning about the research of my colleagues and the many fantastic STEM students that we have here at Berkeley over the course of this year. So my stint will soon be over as interim dean, and our new dean for mathematical and physical sciences, Steve Kahn, will take over in the fall. Steve is an astrophysicist who also does work in the Atacama Desert, as we just heard about from Adrian. And he will work with Mike, continue to work with Mike to bring you more fascinating and inspiring science stories in the year to come. So thank you for joining us today and for supporting the basic science research at Berkeley. Please do reach out if you would like to learn more about anything that we've covered. We'll be sharing additional resources with you by email about today's topics and speakers, and a video of today's session will be available on the website, basicscience.berkeley.edu, about a week from now. You can always return to the Basic Science Lights the Way website, and you can watch episodes for the installments that you've missed going back multiple years, which I can tell you I have done. It's really a fascinating set of topics if you take a look. Finally, we're at the end of the academic year, so I just want to congratulate any of our students who are listening in and are graduating this year. Congratulations to you. This is a very exciting time of the year to see all of our students graduating and moving on to some really exciting careers. So until next fall, stay curious, fiat lux, and go Bears.